All right, so this is the first recorded audio for cell membrane transports. I'm going to make a video for each one of these. I don't know how much we will get to in class. I know that I was supposed to have some of them recorded for when I'm at my conference in just a little while, but with the snow days that are seeming to keep on accumulating, I didn't know what was going to be happening with weather in the next week or so. So I'm just starting at the beginning, and we can use what we need. And if there's stuff we don't need to have videos of, well, then they're there just in case, I guess. So we're going to look at membrane transport. So we're going to look at membrane structure. We're going to look at how diffusion works across that membrane. We're going to look at mediated transport, so things that can't go directly through the membrane itself. We'll look at osmosis, which is the transport of water across the membrane. We'll look at epithelial transports. So we're going to look at a bunch of different types of transports, how things get into and out of the cell. And that's really, really important, as far as the cell goes, because this is how things can get into and out of the cell. And so when you alter permeability or different things like that, that can alter and change the rate at which things can enter and exit the cell, or if they even can enter or exit the cell. So we're going to look at a bunch of different things based on that. So when we look at the membrane structure, so you have to remember that a cell membrane is that phospholipid bilayer, so it's a double layer of phospholipids. And remember, they have those polar heads that face outside and inside of the cell, and then the little lipid tails that are kind of sandwiched in between there. And so certain things can pass right through the phospholipid bilayer, and then other things are going to have to use structures like transport proteins and things like that to move in and out of the cell. So depending on what kind of molecule or an ion or an atom or things like that, that will depend on which kind of route it can use to get into and out of the cell. So excuse me, when we look at this movement of molecules across the cell, membrane is also called transmembrane traffic. So trans is to cross that membrane. So going from that extracellular fluid to the intracellular fluid, so the outside of the cell to the inside of the cell. So when we look at that, the plasma membrane or cell membrane is essentially a barrier. It's kind of like a boundary, like the walls of a building. It separates the inside of the building from the outside of the building, or in the case of the cell, what's outside of the cell from what's inside of the cell. So it is going to allow certain things to pass through and certain things not. So it separates the intercellular fluid from the extracellular fluid, and it's going to kind of regulate what can move in and out. So there's certain things like the nucleus of the cell. You don't want that leaving your cell. So it can't pass through that phospholipid bilayer. It can't pass through a protein channel. And that's a good thing, because we don't want that genetic material that's encased in the nucleus of our cells to be able to just freely pass into and out of the cell. That would be a bad thing. The same thing is, when you're thinking about things outside of the cell, there's things outside of the cell that you don't want inside. And so that plasma membrane is acting as a barrier that kind of regulates what can and can't enter and exit, and the rates and of those things that can actually be passing through that. So that is, like I said, really, really important when we look at that. So we'll be able to kind of see differences in that. Like I said, there's a lot of things that you want to be able to pass in and out of the cell. There's certain kinds of ions that need to move in and out of the cell that are really important for things like nerve conduction and normal cell function. Water, you need to be able to get water into and out of the cell. So there's lots of things that are good that need to go in and out of the cell. And they're going to, like I said, they're going to use different modes to move from one side of the cell, like inside to the outside or outside to the inside of that cell. So when we look at membranes, like I said, they kind of are the regulators there. They are considered selectively permeable. So they're selective. If you're selective, it means that you pick some things and you don't pick other things. So it's going to allow certain things to transport across that plasma membrane, and other things not. So, and when we look at that plasma membrane, there are things that we can do to the plasma membrane. There are different molecules that you could add that could block certain kinds of channels, and that would prevent certain kinds of ions from entering or exiting if those channels are blocked. If you're looking at places like the lungs and you have fluid on top of those membranes, it can basically, cause that membrane to become thicker, and it could, alter the permeability. So there's things that can happen that can either increase or decrease permeability. If you open up a whole bunch of channels, then, that could increase permeability. If you close a bunch of channels, that can decrease permeability. Like I said, or if you add liquids to the surface of a cell, that could decrease permeability. So there's all these things that can alter the permeability of our cells. So we're going to see that in lab when we do the lab with the dialysis tubing with osmosis, we're going to look at the membrane and the permeability, and we're going to see things that are important to that, like surface area. So hopefully now some of these things we're doing in lab are going to start tying into what we're talking about in lecture. All right, so what can be transported through the into and out of the cell? So you can have lipid soluble molecules, if I could say the correct words. So things like carbon dioxide and oxygen, those are lipid soluble, or other lipid based chemicals. Those things can pass directly through the plasma membrane. You could have things that, in those things like that, like oxygen and carbon dioxide, they're nonpolar, they're relatively small, they can pass right through. Since they don't have a charge, they can go right through that plasma membrane. If you had something that was lipid-based, even if it was bigger than those molecules, it could pass through because it's lipid-based. If you have something like an ion, so remember, an ion is a charged particle. It could have a positive charge or a negative charge, but it does carry a charge because it's either lost or gained electrons. So in the example here, it's showing sodium and potassium. Sodium is a positively charged or cation, and that means that it has lost electrons, but it does carry a charge. So now it's considered a polar molecule, so it has a charge. So polar molecules cannot go directly through the plasma membrane because those lipid tails that are kind of sandwiched in between are going to prevent them from being able to pass through there because they are not attracted to that charged particle. So even though they're small, they can't go through that plasma membrane directly through that phospholipid bilayer. They're going to have to use a protein. So they're going to have to use a protein channel to go from one side of the cell to the other. You can also have things like large organic molecules, glucose, amino acids. If you have something that's really big, that's too big to fit through, a channel or a pore, it's going to have to use transports that change the shape of that plasma membrane. So if you guys remember back to anatomy, we talked about things like endocytosis and exocytosis, where the plasma membrane actually, would change shape and engulf things and bring it in, like endocytosis, or you could have like a little vesicle inside of the cell come to the surface of the, inner surface of that plasma membrane, kind of merge with that, and then it could release those substances out of the cell, but it changes the shape of that plasma membrane. So we'll see that there's even some large organic molecules that can get in and out, even if they're too big to go through a channel, or they're not able to go through the phospholipid bilayer. So we're going to look at some of those different kinds of transports. The first thing we're going to look at is diffusion. So we look at diffusion. Basically you have solutes that move from like a high concentration to a low concentration. So they always say that they move down the concentration gradient. So when we look at this, remember the solvent, in this case where we're showing you the little beaker that has water in it, that's going to be, the water is the solvent, that's what's doing the dissolving. If we added a solute, let's say it was sugar, like a sugar cube, that solid little sugar cube, this is a lower amount, that's going to be the the solute. So we had our solvent, the water, the solute, our sugar cube, and then when they combine, you get a solution. So hopefully some of those words are familiar from that chemistry review test. So here we can kind of see when we started here, when we put our solute in, you had a low concentration of solute all over here and a high concentration here, what was all in that little cube. And then because of the movement of molecules, it kind of breaks it apart, and you'll see that it will distribute itself. It'll go from that high concentration, the little sugar cube, it will dissolve and it basically goes into solution. So you get a solution that has an equal distribution of the sugar molecules. Like I said, if that was sugar, it could be salt, it could be all sorts of different things. But when we look at diffusion, it's always going to go down a concentration gradient. It's going to go from high to low concentration. It's not going to require any energy. You don't have to do anything for it to basically go from the high to low concentration. With that, it will just, go from high to low when the random motion of molecules happens, and it kind of breaks that apart. So we will see that. So when we look at simple diffusion, the membrane permeability is influenced by the properties of the membrane and the nature of that diffusing substance. So when we kind of look at that, you are going to see that things are going to go from that high concentration to low concentration, and the properties that are going to allow that is if you have something that, if you're using simple diffusion to get into a cell, it's going to have to be something that is basically lipid soluble, so small, non-charged, that can go from a high concentration to low concentration, either into or out of the cell. So we're going to be looking at those things. So one thing that we kind of need to understand is that the world kind of in the universe all goes towards what's called entropy. So entropy is random motion. Things seem to go towards, when you talk about entropy, they go towards disorder. This is the second law of thermodynamics, and basically you have things that are moving around, and they always go from a high to a low concentration, so it creates more disorder. So when we kind of look at that, they often describe it as things like falling apart. So molecules bounce around and alter the directions, impacting other molecules. So you think about sometimes it's hard to think about in the air. We just kind of don't think about it most of the time, but in our air we have nitrogen gas, we have oxygen gas, we have helium. There's carbon dioxide. There's all sorts of gases in the air, and they're, those little molecules or atoms are bouncing around, bouncing into each other, hitting walls, hitting all sorts of things, hitting into us. They're so small we don't feel them, or we don't see them impacting things, but they are always, bouncing and moving around. So we don't really notice this until we do something like, if, for instance, you spray air freshener. So let's say that I took air freshener, I sprayed it right here. So where I'm spraying the air freshener right now, right here, there's a high concentration of that air freshener. How do I get, if the room doesn't smell good, how does spraying the air freshener right here, if it smells bad in that corner too, how do I get it to not, to get to that back corner? Well, I don't have to sit here and fan it. I don't have to get a fan that is powered by electricity to move it around. It naturally is going to move from this high area, of air freshener to the low area of air freshener. Because you have all these gas molecules that are moving around, bouncing into each other. They're going to help to distribute that, and it's going to go from a high concentration out to where there is no air freshener to low concentrations until it's kind of evenly distributed through our room. So things go towards disorder. So here we can kind of see, here's one molecule bounces into another one. So here's the department of entropy, and so it looks like everything's kind of falling apart. Like I said, things tend to go towards disorder. So when we think about disorder, and you kind of think about that, think about, you do your laundry. So you do your laundry, you wash clothes, you dry your clothes, you put all your clothes in the laundry basket. how much easier would it be to take that basket of laundry? Because you know that after you get it, you have to take it and you have to fold up the shirts and match the socks and do all of that. And then you go and you take it to your room and you open your chest of drawers and you put the clothes into the proper drawer. That, you know, that takes a lot of energy to do, you know, and kind of compartmentalize it and put it into this nice organized manner. It would be much easier to just take our laundry basket and go to our room and just take it and heave it and let the clothes fly wherever they wanted to go. Things tend in nature to go towards entropy. It doesn't require energy. It's the natural state of things. Like I said, and we can see it just by thinking about the air freshener. But like I said, sometimes it helps if you think about, like, you know, it takes a lot of work to get everything like packaged up and folded up and put it into the drawers versus just flinging it wherever. So things to go towards that entropy. So this is the natural state of things. We also have what's called Brownian motion. So Brownian motion is going to be the movement of larger molecules when smaller molecules kind of bounce against them. They're not truly moving themselves, like you'd see with like things like bacteria that have flagella, or like a sperm cell that has flagella, or you know, it's not truly moving. So we are going to soon, hopefully, be able to do a lab that looks at osmosis using red blood cells. And you guys, when you were in anatomy, you looked at the red blood cells on a slide, and the slides were all pre-made and they were fixed. So when you put that slide onto your microscope and you look through it, all the little red blood cells and the white blood cells, they all just stayed still. They were stuck down. You know, there was glue involved and held a little cover plate on there, and none of the cells were moving. When we do our lab, we're going to see that you're going to take a sample of blood, you're going to add some different solutions, you're going to see how it affects those cells, but you're going to look at these red blood cells under the microscope. And it doesn't matter which one we're looking at, a hypertonic, hypotonic, isotonic solution. It doesn't matter which one you put under there. You put your little blood and different solution, isotonic solution maybe, on the slide. You're going to put a cover slip on there. You're going to look at it under the microscope. When you put it under the microscope, something weird is going to happen, or it's going to seem weird, because when you looked at the pre-made blood slides, everything just stayed still. You're going to look at the blood slide that you just made from that fresh blood, and you're going to look at it, and the cells are going to be like moving on there, like kind of like streaming. Usually they're kind of almost like they're flowing, like streaming past you. And it kind of sometimes weirds people out. They're like, why are they moving? Like, you know, because they're red blood cells. They don't have flagella. They don't have anything that's moving them. They're not being pumped through an artery or, you know, being pushed through a vein anymore. So why are they moving around? Well, when you put them onto that slide, you have little water molecules that are bouncing into those red blood cells, you know, because the random motion of that's in that fluid. And so those red blood cells in turn move. And so it looks like they're just moving all over the place. But it's through Brownian motion. So, you know, if you have something small that hits it, it can bounce that and move that. And like I said, when we look at those red blood cells on that, and eventually it kind of slows down, you know, a little bit. But when you first put it on there, a lot of times you're like watching it and you're like, oh, they're just like streaming by, and people are kind of taken aback because they're used to all the slides being fixed like you saw in anatomy. So Brownian motion is movement of large particles, because we have all these little particles that are moving around that bounce into them and kind of, you know, push them or move them around. So when we look at diffusion, there are going to be some different factors that are going to affect diffusion, that are going to impact diffusion. Is it going to increase or decrease it based on what happens? So we're going to look at some 6 different things that are going to impact diffusion, when we look at cells. So the first thing we're going to look at is temperature. So when you increase the temperature, you're going to increase molecular activity. This has an impact on immune function that we're going to kind of look at. But, you know, when we look at this, if you think about, like, you know, on a cold morning, get up and you don't want to move very fast. You know, but then once you've been up and you're moving around, you feel warm, you know, it's easier to move around. Or, like, you know, same thing when you exercise, usually they warm you up. They start you out by doing activity that's not as strenuous. And then once you've warmed up those muscles, then they're going to really get you into more of an intense workout. So you're going to increase the molecular activity when you increase the temperature. Molecules move faster when they are warmed up. So think about if you have, let's say that we have 3 teacups of tea. One of them, we have tea that's we made and we put in the refrigerator, so it's pretty cold. Maybe we just have some room temperature tea. We left tea out on the counter, so it's room temperature. And then we have some tea that we've just made, hot water. We just made that with a tea, like a tea bag. So we have these 3 cups of tea. So a cold one, room temperature, and hot. Let's say that we are going to put sugar in our tea. So we're going to put, do you guys know what a sugar cube is? You guys, I don't know. They used to be, I think, more popular, like you'd go to the bank and they would have it out with like the coffee station. And if you were good, your parents would be like, you can have a sugar cube. It was like a candy, but it was just pure sugar in a little block. So imagine our 3 cups of tea, and I'm going to put a sugar cube into each one of those cups of tea. So we're not going to stir them. We're just going to put our little sugar cube into each of those. Eventually, we're going to see that the sugar in all of the 3 cups of tea is going to dissolve. But which one is going to dissolve the fastest? So, probably most of you, without even thinking about it, you're going to say the one that's hot. Hot dissolves faster. Well, why does the sugar dissolve faster in the hot? It's the same substance. It's tea in all 3 of our cups. Why does it dissolve faster in the hot cup of tea? Well, the molecules in that liquid are moving much faster. When they're moving faster, they're bouncing against that sugar cube more often, more frequently. And that's going to break, down the little sugar cube, and it's going to go from the high concentration of sugar to the low concentration, the tea that doesn't have any sugar, and it's going to break that down and kind of distribute evenly at a much faster rate because the molecules in the hot tea are moving around much faster. And then if we look at the room temperature tea, it would dissolve, faster than the cold tea. And that's because when you go down in temperature, you're going to see that activity level decreases. The molecules are going to be moving more slowly. If they're moving more slowly, the amount of times they're going to randomly bounce into that sugar cube is going to decrease as the temperature decreases because they're moving more slowly. And so it's going to take longer to dissolve that sugar cube. So we can kind of see that. So hopefully that makes sense. So when we look at, we're talking about here, it says has an impact on immune function. So a lot of times if you have, like as a kid, when you went outside, your parents would tell you to put on a coat, put on a hat, and gloves or mittens or a scarf or whatnot, when it's cold out. You'll catch a cold if you don't put on a coat. You don't catch a cold when you go out. You could go out in a t-shirt and it doesn't cause you to being, out without a cold doesn't cause you to be sick. What happens is that when you go out in the cold, you suppress your immune system. Your immune system the molecules are moving slower. Your immune function is lower. So you depress your immune system when you're in the cold, and your body becomes cold because you're not dressed properly. And so you're more likely to have, to get sick because your immune system isn't functioning at the same level as when your body's at its normal core body temperature. On, kind of the flip side of that is you do get an infection. Why does your body reset your temperature? Why do you get a fever? The fever is your body causes a fever. It basically sends chemicals that are going to go reset your hypothalamus to say, instead of being at your normal 98.6 degrees, that's your normal body temperature. Some people run a little hotter or a little colder, but we'll just use that as the temperature that they always say is your normal body temperature. Why does your hypothalamus say your new body temperature should be 102? Well, once again, if you increase that temperature, those molecules are going to be moving faster. You're going to improve how quickly those cells can work in your immune system. So it can have an effect on your immune function. So here you can see when it got hot in the fire, that little molecule bounced around like crazy because it warmed up. So we can kind of see that. So it will, when we look at all these things and we talk about cells, we talk about molecules and different things like that, you have to understand that when they're moving around, it's all random. They are not, like I said, when you talk about different ions moving around, going from high to low concentration, we talk about things moving like this, it's all just random motion. It's not targeted to one particular cell. Like when we talk about the endocrine system releasing hormones into the bloodstream, when they get to those target cells, how do they get there? It's not like there's a homing beacon that says, hey, come here. They get dumped in the blood and then they go places, and eventually, hopefully, they hit those targets. So a lot of this, when we look at this, it's random motion. There's not anything that has to do with, it has to go to this specific thing. Like if we want to go to a specific place, we say, well, I'm going to go to the cafeteria, and then I walk there. Or if I didn't know how to get there, I would ask people. But when we look at these little ions, molecules, and things like that, they are moving in random ways. They just bounce off of things, and hopefully they run into the right thing. So the same thing with like your immune system. If you're looking at antibodies and different things like that, the faster they're moving around, the more likely they are to bump into the foreign particles, so they can, tag that as being foreign. So we'll see that. The second thing we're going to look at is molecular size. Particles with a smaller mass have increased entropy. We saw this when we were in lab. So hopefully by the time everybody watches this, everybody will have completed that first diffusion lab with the crystals. As of right now, I think I have 2 lab sections that still haven't done it because we keep on having snow days. But if you have a smaller mass, you're going to move more rapidly. If you move more rapidly, you're going to be increasing that entropy because you're bouncing off of things and creating more disorder. If you're bigger, then there's less entropy because the molecule is moving more slowly. And this is at any, if you're looking at the same temperature, these particles being at the same temperature. So here we can see our little molecule moving around really fast. And then our big molecule here goes lumbering along there. It's much slower. So the bigger the mass, the slower it's going to go, and the less entropy you're going to see because it's moving around more slowly. The smaller it is, the more it bounces around, the more it hits into things, the more it bounces this way and that, and it creates more disorder. So when we look at the size of molecules, why, once again, a lot of times people are like, okay, you told me size matters. Why do I care? How does it affect the cells? So it can do things like impact respiratory gases. So when we look at helium, helium is smaller than nitrogen. So if you think about our air in here, usually when we think about air, we think, oh, it's the same as oxygen, because what do we pull from the air that's really important to us? Oxygen. But really only about 20-ish% of the air that you have in the, the room around you is oxygen. A great percentage, a much greater percentage, is nitrogen. You have some helium in there as well, carbon dioxide, you have these other gases. But when we look at this, if you have ever been scuba diving, they strap the tank to you and it has oxygen in it, of course. But they also put helium in it because it's easier as you go down and the pressure increases, it's easier to breathe that helium because it doesn't become as viscous with that. And it basically, when you look at nitrogen, sometimes when that comes out, it can collect in tissue spaces and kind of push oxygen out, and you can get what's called the bends. So it comes out of those tissues faster, the helium does, and it's going to prevent the bends. So now when you have tanks full of oxygen, they also incorporate helium into that so that it can be easier to breathe under those deep pressures, and also is not going to cause things like the bends to happen when you ascend, from a low pressure to normal pressure as you come up, from a deep sea dive up towards more like sea level. So here's our little experiment with the crystals. So if we looked at that, and it kind of shows you here is going through and doing that diffusion. So this would be our little auger plate where the crystal is diffusing. And this one says, which crystal has the lower molecular weight? So remember, lower molecular weight. So if it's lighter, is it going to diffuse more or less? So remember, the light one can move more easily. So that was my example was it's kind of like having the bowling ball and the boulder. Which one, if you had a minute to push them, which one is going to be able to move farther? So that kind of acts like our diffusion. Well, the bowling ball, you can pull it, we can roll that down the hall really easily. Our boulder, let's say that it was a perfectly round boulder, so you should be able to push it. It's not all jagged, but it's massive. How far are you going to be able to push it? Not very far. So when we look at this, the one that has the lower molecular weight is the one that had the greater diffusion. So it would be this kind of magenta, purpley looking one here versus the blue that we're seeing here. The nature of the media also has a lot to do with diffusion. So when we look at what kind of media we have, how fast something can diffuse is going to be altered. So remember our states, we can have liquids, we can have solids, and we can have gases. So when we did our diffusion experiment, we used agar. And agar, like I said, is kind of like a firm jello. So it's like a semi-solid. It's a solid, like you could touch it and it's a solid. It's definitely not a liquid. But it's not exactly the same as like, the table, like here, when I hit the table here, like I can press on the table really hard and I'm never gonna go through the surface of that table. So when we look at those different types of media, we're gonna see that diffusion is going to happen more rapidly in thinner media. So solids, are the thickest, they have the most structure to them, and then your semi-solids, like your agar. Water is thinner than, a semi-solid, and then air is much thinner than even the water. So if we looked at our little crystals, if we put our, we had the crystal, we had the potassium permanganate. If we took a crystal potassium permanganate, and I have a petri dish here, and I put a potassium permanganate crystal on there, I have an equal size crystal of potassium permanganate, and I drop it into an equal amount of water, like in a little petri dish, and we observe them. You would have seen that, that in 30 minutes, your potassium permanganate, it might have moved. I think numbers upwards of like 12 or 13 millimeters, and that was in 30 minutes. So 30 minutes, it moved like this much. When we drop that same size crystal into our little petri dish of water, you would see that within a few minutes, maybe not even a minute, that little crystal would have diffused. You would see that color radiating out and diffusing out into the water, and it would have reached the edges of your little petri dish within a minute or two. It would have happened pretty rapidly because the water is a much thinner type of media. So you can kind of see the water, our little molecule here was bouncing around really rapidly in the water, versus when it's in that semi-solid, like the agar or jello, it's going to move much slower because that medium is thicker. If you think about gases, now I can't really put a crystal of potassium permanganate in it. We can't watch it diffuse through the air, but you can think about how quickly diffusion happens with something like air freshener. So imagine, 30 minutes with the crystal in the agar, it takes 30 minutes to move like 13 millimeters. Crystal into the water in a petri dish, let's say 2 minutes. How about if we were all sitting in lab? So imagine you're sitting in lab right now, and I spray air freshener in the room. So I spray right up here by the desk. Within a couple seconds, people in the first row are like, oh yeah, I can smell it. And then maybe 5 seconds later, the people behind them can smell it. And usually within probably 30, 40 seconds, the people at the back corners of the room, they're going to smell that. So it's diffusing very rapidly. That air is much thinner than even the water. So it's going to diffuse very rapidly. So you can kind of see that the how thin or thick, how easily is that those molecules can pass through that media. That will be based on the thickness of that. So hopefully by thinking about like your semi-solid, a liquid, and a gas, those examples will help help you make sense of that, and it kind of make it understandable. All right, so diffusion limits cell size. So I kind of alluded to this when I talked about the diffusion lab with the crystals. When we look around, we don't see, we have billions of cells, like especially if you start including all the bacterial cells that are in us. So, we have all of our human cells, but we have bacterial cells and different things, in our bodies too. But we are a big organism, but we're made of lots and lots and lots and lots of cells. When you think about other organisms, like single-celled organisms, do you ever see single-celled organisms at the size of like your dog or your cat? No, that's too big. Basically, diffusion really limits the size of cells. They can't get too big because it would take too long for nutrients and gases to diffuse into and out of the cell and allow for those activities to happen. So single cells can't get massive. You don't see baseball-sized cells. You don't see cat-sized cells, single cells. When you look at single-celled organisms, you have to look at them under the microscope because they're small. They can't get very big. Cells can't be very big by nature because diffusion, the bigger that cell gets, the more inefficient diffusion is. So it's going to limit our cell size. So, with that, media molecules diffuse more rapidly in gases than in liquids. And like I said, if you think about the air freshener, that moving around, it's going to diffuse that relatively rapidly. Like I said, you could figure out the rates. Like I said, unfortunately, we can't use the crystals for all three of them. But like I said, if you think about air freshener and how quickly it would disperse through a whole room, it's very rapid because the gas is very thin. So those collisions are going to, going to be more frequent than in a liquid than in a semi-solid. So like your collisions, the encounters, because like I said, all those molecules are in random motion as you go from a gas to a liquid and then a liquid to a solid. Those encounters, those collisions are going to decrease, and that's going to make those interactions go down, and that's going to make the diffusion happen less rapidly. So diffusion happens most quickly in gases, then a little bit slower, but still have more. A little slower than your gases is going to be your liquids, and then the slowest one will be like your semi-solids, like I said, agar or jello, something like that. Concentration. So our fourth thing that's going to affect diffusion is concentration. So diffusion always goes high concentration to low concentration until the concentrations become equal, and then they all just kind of bounce around. So if you had, like a beaker that had like a semi-permeable membrane in it, and you had, water on each side, and then you put like, I don't know, salt in there, and the salt could travel through your semi-permeable membrane. It would go from the high concentration salt to the low concentration of salt until it got to equilibrium, and then it would randomly, for everyone that went in one direction, one would go the other direction. So they move around, they go from the high to the low concentration until it gets to that equilibrium. Once they get to equilibrium, it doesn't stop. It just means that there's no directional flow at that point. But we'll get to that in a minute before I get too far into that. And once again, it's all dependent on those molecules bumping against each other. And when they get bumped into, it changes directions, and then other molecules bump in, and then it goes from a high concentration to the low concentration until you get to that equilibrium. Like I said, the molecules do not have feelings. They don't want to go from high to low concentration. It just is the natural tendency of things. Everything goes towards entropy. It goes towards disorder. Having that sugar cube or a salt cube, that's ordered. having all of it packed into one little area. That's a lot more order than having it all distributed through a beaker full of liquid. So things tend to go towards disorder. So here we can kind of see over time, solute molecules that are going to a solvent are going to evenly distribute themselves. So they go from high concentration to a low concentration until you have eventually you've gone to equilibrium where you have the same concentration everywhere. So higher concentration molecules randomly moves towards a lower concentration. Like I said, it was all based on there's, that could be salt, it could be sugar, but there's water molecules in here bouncing and hitting those, and they basically bounce and hit into each other until everything is evenly distributed. Are things still moving in here? Yes, those molecules are still moving in there, but there's just no net movement at that point. So once it gets to this state where you have an equal distribution, that's going to be that steady state. So it goes from high to low concentration until you get to that steady state. So here we can kind of see the next one. Here, this is like little compartments. So this is showing you high concentration, whatever the purple little dots are there. So in compartment 1, lots of purple dots. In compartment 2, no purple dots. So as long as this membrane that separates the two compartments is permeable to the purple dots, like I said, it could be salt, it could be sugar, it could be whatever, as long as it's permeable, they're going to move from the high concentration to the low concentration. So here we can see, when you go from time A to time B, now you can see that the natural tendency is to have the little purple dots move from the high concentration to low concentration. You'll also notice there's an arrow now that we have some purple dots over here, there's a little arrow that's showing that things can go this way. Why would a molecule want to go that way? Remember, molecules they don't care. They're all just random motions. So if you have a molecule over here and it happens to bump into this semi-permeable membrane, it can go back over here. It's just that the likelihood is lower. So the arrow is less here because there's a lot more purple molecules on this side. And them randomly going over here is more likely to happen running from 1 to 2 than from 2 to 1. Eventually you'll get where they've gotten to that steady state, that state of equilibrium where you have the same amount of that purple molecule on each side. And then you'll notice that the arrows don't go away. The arrows are equal. They're equal on each side. And that's because at equilibrium, things are still moving. Those molecules can still pass through that membrane. It's just that they're passing through in the same number. So every one that goes over here, there's another one coming over here because it's equally likely at that point because the concentration is equal on both sides, which is kind of hard to think. We always tend to think once it reaches equilibrium, everything just stops, but it really doesn't stop. They're still moving around. So this is just a graphic kind of showing you. So if the picture doesn't work for you, we have more maybe like a diagrammatic math brain. This kind of shows you the green line's compartment 1, the purple line is compartment 2. So here we can kind of see that at the beginning here in compartment 2, there was 0 of the little purple molecules. There was all 20 of them were over here. And then as time passed, you can see that the concentration is going down in compartment 1, and the concentration in compartment 2 is going up until you reach that equilibrium where you have the same amount on each side. So like I said, depending on how your brain works, sometimes one picture is better than another for you. So I try to include some different, ones. So if you, kind of have more of a math brain, hopefully that works. Or maybe you work more visually where you can see little individual molecules kind of represented as those dots. So hopefully that is helpful there. All right, so next thing we're going to look at is flux. Flux is also called the flow. So if you're looking at this, when you see flux, you could, that's also a kind of equivalent to flow. Sorry, my handwriting looks less than stellar when I draw with the computer. So flux equals flow. So it's the amount of a substance that's crossing the surface per unit of time. So probably like the speed that it moves there. So All right, so I'm gonna go right back. No, I don't want that. Sorry, guys. One day I'll figure out how to get rid of that without exiting out. All right, so if you double the concentration, so let's say if you imagine your little compartments, you had side A and side B. If you double the concentration in side A, you're going to basically double the flow. So you're going to increase the rate at which those little purple dots are moving from compartment A to compartment B. So if you went from having 20 of them over there and 0 on side B, and then you increased it to 40, so you went from 20 to 40, you doubled that, it's going to move, those molecules are going to move twice as quickly to kind of get to that equilibrium state there. So kind of like the rate of speed in an automobile. So it's going to be moles per second for that particular molecule, whether it's oxygen or carbon dioxide. If you're looking at gases, if you're looking at compounds, it could be glucose, how many glucose, the moles per second of glucose molecules, or sodium ions, or whatever it happens to be. But flux is flow. So the amount of time, it's basically like speed, how quickly they go from one place to the other. And like I said, if you increase the concentration, you're going to increase the flux. If you decrease the concentration, you're going to decrease the flux. They are directly proportional. So here we can kind of see, here's two compartments. So we have a side A, well, this is our side 1 and side 2, and this is compartment, our example 1, and then here's our example, I should say A. So this is A and this is B. So here we can see on this one we have 4 molecules on side 1, and then if we counted these, 1, 2, 3, 4, 5, 6, 7, 8. We have 8 molecules, so A has twice as many as B. And so we know that we have, here's our little semi-permeable membrane. It's the same substance, the little lime green dots there. So our little membrane there is permeable to whatever the lime green dot represents. And we can kind of look, if we were looking at this and looking how quickly it went from side A to side B, we could see, they bounced into each other. So if we time that, it usually takes, it's going to take longer to, get, to go, if we had double the concentration here, it's going to more quickly go from side A to side B, because they're more likely, think about them hitting into each other and then bouncing against sides to hit that semi-permeable membrane to move to the other side. So a lot of times what I kind of tell people to think about is, imagine that we are in like our classroom, but imagine that we can take all the benches out of here. So imagine we're all in here and it's just a big empty room. So imagine that I have 10 students in here, so one of my smaller sections. And I tell you guys that you can walk in straight lines until you bump into the sides of the wall, or the board or whatever, or into each other. But anytime that you bump into each other or into the wall, you're going to change directions. So if we, and we can only go out the door that we normally come in and out of. So how long would it take if we had 10 people in this room and we're all walking kind of in straight lines until we bump into another person or a wall? And then eventually we'll hopefully get in the right direction that we are walking in a straight line and we can exit the room. It might take quite a bit of time. Now imagine that we double that to 20 people in the room. How much more likely are you going to bump into someone if now there are 20 people in the room instead of 10? Or we could double it further and we have 40 people in here. So every time that we double it, you're much more likely to bump into people because now we're closer together. And if we bump into each other, we change directions. The more time we change directions, the more time that hopefully we're going to get oriented into the right direction to move out of the door. So, you can see that as you increase that concentration, you're going to increase the interactions, the random motion that they're bouncing into each other and into the walls, and they're more likely to, orient themselves into a direction that's going to cause them to go across that semi-permeable membrane. So we can kind of see that. So sometimes, like, I try to make you guys part of the experiment, even though We're not actually here and doing it, but you can picture that in your head. You can see, you know, oh, I can envision that, what that would look like, and then increasing the number, you know, of people there. So here we can kind of see, remember, flux is flow. So over here, this is our semi-permeable membrane right here, the little gray line. Here's compartment 1, it's a high solute concentration. You can see all those dots are all tightly packed together. Over here, this is compartment 2, has low solute concentration. And so we can see that you can have, the natural tendency would be to go high to low. But once again, these are just randomly moving particles. So when you know, the likelihood that these guys are bouncing into each other and bouncing over here and going into this low concentration area, much higher than the reverse, but there are still particles that could bounce each other over here and cross back into this high concentration area. But you'll notice that we have a large one-way flux going from the high concentration to the low concentration. We have a small one-way flux that goes from the low to the high concentration. However, when we look at the net flux, so the net flow or net movement of those ions or molecules, it's going to go from the high concentration to the low concentration. And that would happen, and that would be true. We would see this net flux going from high to low until it reached equilibrium. And then, like I said, once it gets to that equilibrium where it's equal on each side, then you would see, you know, those lines, those one-way fluxes would end up being the same because there's no net flow there, because they are now equal in concentration. So that net flux or net flow is the difference between two one-way fluxes. The movement in one direction is greater than the other. Like I said, you know, it's going to go like here, high to low is that one-way flux is much larger, because if you think about these bouncing into each other and crossing that semi-permeable membrane there, that's much more likely to happen than the reverse that we're seeing right here. So then the overall direction we see that these molecules are moving, or ions, whichever they are, is in this direction from the high concentration to the low concentration. That would be our net flux or net flow. All right, so our next thing that we're going to look at is going to be basically lipid solubility. So, you know, like lipid solubility, why do I care about lipids? So remember our phospholipid bilayer. Things that are lipid permeable, things that, you know, are made of lipids or are nonpolar, are going to be able to pass right through that plasma membrane. So if you're lipid based or nonpolar, you're going to be able to pass through that phospholipid bilayer. So you're going to have, excuse me, that you're going to care about the membrane permeability of lipid based molecules. So experimentally, they have gone through and determined the permeability of the cell's membrane to different kinds of molecules, ions, different sorts of things. And basically, the higher the permeability means the higher rate of transport. Here we can see, you know, if you had something like potassium, if it was an ion, it would be a charged particle. It'd have to go through a protein channel. If you had something like oxygen that's nonpolar, it could go right through that phospholipid bilayer. So membrane permeability constant, that's the Kp, describes the pore size of the membrane. And like I said, it's experimentally determined. So if you have membranes have a high P value, they have large spaces. If you have a membrane with low P values, it has small spaces. So the molecule's lipid solubility, the molecular weight, temperature, and membrane characteristics are all going to affect that membrane permeability. And we're going to see in just a minute an equation, so then everybody gets like, oh no, we have an equation, we have to, you know, solve equations. No, you're not going to have to solve math. It's just a kind of way to relate this to you. So if you have large spaces, these high P value are going to be lipid soluble. If you have a low P value, though it's like small spaces, that would be not be lipid soluble when we look at that. All right, so the flux equation, so like I said, don't get worried. A lot of times people start seeing the letters and things equal this and that, and then they get real worried that you won't have to do math. It's just kind of relate this to you with that. So when we look at the flux equation, one big factor is the surface area, which is denoted as A. So that's the surface area available for diffusion. When we look at that, we're going to look at concentration differences. So basically, the concentration of basically what's going out versus what's going into the cell. So CO, so concentration of what's moving out minus concentration of what's moving in. And we also have efflux and influx, which is the movement out of the cell. So F sub E is what's moving out of the cell. So efflux. And then the movement into the cell is the influx, that's F sub I there. So if we look at this influx, we would look at the membrane, we're basically looking at that membrane permeability for a particular substance, times the area, and then the concentration of what's moving out. And then the efflux, we look at that membrane, once again, the membrane permeability, the area, and the concentration in. And then the net flux would be this is kind of constant, like your constant, basically the permeability constant times the area. And then you, instead of having two separate equations, you can combine what's moving into and out of the cell. You subtract those and you can kind of see that. What you want to, like I said, I'm not going to make you do math, so I'm not going to give you an area and a KP value and a CO and a CI value and then ask you what the next net flux is. I'm not going to do that. If you took it's in Gen Chem 2, I think. I know I've done, you have equations like this, so you have to do, I think it's Gen Chem 2. I can't remember. I'm pretty sure it's 2. Maybe it was quantitative analysis I did that, and I don't remember, but you can do the math for this with that. But what I do want you to take away, if the net flux is negative, stuff is going to be moving out of the cell. If it's positive, things would be moving into the cell. So like I said, you don't have to do the math, but if we had a net flux that was negative, that means things are moving out of the cell. If you have a net flux that's positive, that means that is moving into the cell. Because if you did the math, because obviously, if you do your subtraction, you get a negative value, that's going to tell you it's moving out. And then if it's positive, it's moving into the cell. So the direction and magnitude of the flux. So remember, tendency of molecules to spontaneously move. Like I said, the molecules are doing this naturally. They are bumping into each other, the entropy going from order to disorder. So it's always going to go high to low concentration. That's a natural tendency of things. And the larger that concentration difference, the greater that flux or flow. So we would kind of see that. Trying to look and see. Trying to make sure I don't make this too long before I stop and give you guys a break. So passive transport. So when we talk about something being a passive process, passive means that it does not require energy. So it's a natural tendency of things to go from a high concentration to a low concentration. It's going to happen spontaneously. It's just that entropy going from a more ordered state to a more disordered state. So high to low concentration. It doesn't require any energy. So think about if you have a 2-year-old, or maybe you've watched a 2-year-old if you don't have your own 2-year-old or haven't had your own 2-year-old. If you've ever watched a 2-year-old, think about if they have, toys and stuff in their room, and you come in the room, it's all nice and organized. You leave for 5 minutes and it looks like a tornado went through their room. They took all of that nice, organized, everything in its place kind of thing, and it went everywhere. It went to disorder, it went to entropy. It did not take any time, really think about their attitude towards this. It didn't take them long. They, it didn't require any energy on the, I mean, they did move, so it used energy, but didn't require an input of energy to go from that organized to disorder. Think about making them pick it up. That's like the worst thing ever. Like, cleaning it up, it takes a lot of energy, a lot of redirecting to get them to put all of the things back into their places, so things naturally go towards disorder. So it's passive process goes from high to low concentration. They occur spontaneously and doesn't require energy. So a lot of times they show like the rock on the hill. If you have the rock at the top of the hill, it will naturally roll down. And as it rolls down, it converts that potential energy into kinetic energy, but it didn't require, it's releasing energy. It didn't require energy to be put into that. If you had to roll the rock up the hill, that would be now not a passive process because it would require energy for you to push it up the hill. So magnitude of that flux or flow in simple diffusion, basically the net flux or net flow is directly proportional to the size of the concentration gradient. So the bigger the concentration gradient, the bigger the magnitude of flux or flow. So it would be linear, like you'd get a linear relationship. So like if you keep on doubling the concentration, you're going to see that magnitude or flux of that flux or flow is going to also double. So here we can see if we had none and we doubled that, you can see that the flux or flow, that net flux or flow is increasing in the same proportion. If I doubled here, we would double, like if we had values over here, we would have doubled the flux or flow. And then if we, end up, you kept on doubling or kept on increasing it, you would see that flux or flow would increase proportionally based on how much you are increasing the concentration. So you can see it kind of is going up in a straight line because they're directly proportional. It has that linear relationship there. So membrane permeability, the more lipid soluble a solute is, the higher the p-value, the higher the permeability, and the greater the flux or flow of that particular substance is. So when we look at things that are going right through that plasma membrane, oxygen, small and uncharged, it can pass through really easily. Carbon dioxide, also small and uncharged, can pass through easily. Fatty acids, steroids, those are both much bigger, but they are lipid based, so they can once again pass through that plasma membrane there. We can determine, doing an oil-water partition coefficient, the ability of a substance to move from being dissolved in water to being dissolved into lipids, using basically it's like a little separatory funnel that we're going to see in a minute. And when we look at the coefficient of how much of that particular substance is dissolved in the water and dissolved in the lipid, you can determine, that what it dissolves in better, the oil or the water. Oh, excuse me. See, this is what happens. I'm talking to an empty room now. I'm getting tired. It's also really cold in here today. All right, so this is our little separatory funnel. I don't know if you probably never used one in chemistry. You did probably use the little burette tube with the stopcock on it. You can kind of see that it has that little stopcock where you can turn that and open up this so you can release the substance there. So you would, in your little separatory funnel, you'd put in water, you'd put in oil, you would put in your substance, you would shake it up and you'd let it sit. And that, if you've ever had like a wave bottle that you made with oil and water, you can take it, you pour the water in, you pour your oil in, you could shake it up. I'll put the cap on first, shake it up, and then that if you set it down and you leave it sitting there, it's going to separate into the water layer on the bottom and the oil layer on the top. The oil kind of floats on top of the water. Same thing would happen here. You'd add your water, you add your oil, you'd add your substance, and then you'd shake it up so that it all mixed together, and then you would let it settle out. So you're going to let that sit. So here we can kind of see, we have substance in here. Like I said, you add the substance, you shake it up, you let it settle, and Then you're gonna drain off those two layers. Like I said, this all looks kind of blue. Like I said, a lot of times we can, depending on what kind of color your oil is, or if you stained your water, you could tell the difference between those two layers. You can see in this little picture, obviously you can see the molecules. You know that like when you're actually looking at oil and water, if you have dissolved things into them, you would not be able to see the molecules and just look at them and count them. Ooh, did not mean to do that. Okay, sorry. So once it's separated, you're gonna drain the water layer. So you'd open the little stopcock here, and you drain the water into this beaker, and then you'd close the stopcock, and then you put a new beaker under there, and you would drain the oil layer in there. And then, so here it's just showing, I only have so much control with this. So here we're separating that out. You wouldn't be able to just pour the little molecules out. It would actually give you the water in each of those, the water here and the oil here. So when you looked at these, if you had a high coefficient of that solute in the oil, that would make it a high lipid solubility. If you had a low coefficient in the oil, then it would have a low lipid solubility. So you basically look at how much was dissolved in your oil versus your water. So here, like I said, when we look at this in pictures, we can kind of see, okay, we counted the two, little molecules here. So we had two, and then in the water, we had four. So it was a 2 to 4 or a 1 to 2. So this tells us when we look at our two little coefficients here, we have twice as many dissolved in the water as we do in the oil. So this would have basically a low lipid solubility. Some things did dissolve into it, but not as much as it dissolved in the water. If it was reversed and we saw 4 in the oil and only 2 in the water, then it would have had a high coefficient for oil versus water. So it just depends. Like I said, it's an experimentally found thing. And so it basically tells you how soluble any particular solute is in oil versus water, so that you could kind of tell. And a lot of times people ask, once again, why do we care? Well, if you're making medication, you want to make sure that it can get into the cells, and if you make your medication lipid soluble, it can go right through that plasma membrane instead of having to go through a channel. So that can increase its ability to get into and out of to go into the cell where that is needed. So we can kind of see this. So an example of this is a real life example is aspirin. So aspirin is acetylsalicylic acid, and so acids have hydrogen. So here, this is our acetylsalicylic acid. I don't try to say it too quickly. And when you put it into water, this acetylsalicylic acid will separate into hydrogen ions and the other portion there, because hydrogen is what makes it acidic, but you get basically ions. You have positive and negative ion with that. And when we look at that, ions are charged particles. Ions cannot pass through that plasma membrane. They cannot go through there because they're charged. They can't go through that little lipid bilayer. So they're not going to be able to pass through there. So we can kind of use that to kind of see, how we could best get aspirin to be absorbed. So when you put, like I said, when you put aspirin into solution, it ionizes. It's going to make the hydrogen ion and your acetylsalicylic ion as well. And when it's ionic, it's going to be impermeable. It's not going to be able to go into the cells. So it's not lipid soluble then. It's too large to go through any pore. So if you're trying to get this into your cells, and it ionizes and you get hydrogen ions and your acetylsalicylic ion there, it doesn't do your cells any good because it's can't go through, when it ionizes, it can't go through the plasma membrane, and there's no pore that's big enough for it to go through. So how would you get it into your cells? When it's in its unionized form, where the hydrogen is attached to the acetylsalicylic portion there, and it forms the acetylsalicylic acid, so it's an acid now, when that non-polar forms, since it's non-polar, it's able to pass through that cell membrane. So, it can go through. So how do you keep it from ionizing? Because you think, okay, you take aspirin and then you swallow it, you're taking it with water, it's going into, what's inside your body cavities, body fluids, a lot of water. So it's naturally, you think, oh, it's going to dissociate, it's going to go into that ionized form when it dissolves there. So how do we get it to stay as the combined, where it's not ionizing? So according to the law of mass action, or Le Chatelier's principle, I don't know if any of you guys in intro chem, I don't think they do Le Chatelier's principle. If you took Gen Chem, you may have heard of that, but basically Le Chatelier's principle is that, if you increase the concentration of something on one side, it will drive it to the other side, so you get balance. So it's kind of like a teeter-totter. If you have one kid of equal weight on each side, you're balanced, but if you put two kids on this side now, you're in balance. So when you put, if you want to drive this reaction to this side where you have the acetylsalicylic acid all combined and not ionized, you need to have that in an acidic environment. Because if you have an acidic environment, there's going to be lots of free floating hydrogen ions. So if you ionize this and you have a lot of hydrogen ions already over here, it's going to make those combine and push back so that you can get to equilibrium there. So when we look at aspirin, where's the best place for it to absorb? It absorbs best in the stomach. The stomach is a highly acidic environment. There's lots of hydrogen ions, just free floating hydrogen ions in the stomach. So if you have like an area where there's already a whole bunch of hydrogen ions, and you swallow that aspirin and it starts to dissociate, it starts to break into hydrogen ions and your acetylsalicylic ion there. But then when you have all those hydrogen ions in that highly acidic stomach, it then forces basically this to go back to do that reverse reaction there, and it's going to make these two combine back into that. And when you have it in this acetylsalicylic acid form, this molecule can actually enter into the cells. So the best place for aspirin to be absorbed is in the stomach. So not in the intestines. The intestines are not acidic like the stomach is. So, when you're looking at these, some of these examples, and you kind of see them, you're like, why do we care? Why do we do things with the separatory funnel? Why do we care if it's lipid soluble or water soluble? What difference does it make for us? If you are bringing something to, market, as far as like medication, where do you want it to be absorbed? Where is it in the format that can be absorbed by the cells? Do you have something that you need absorbed in the intestines? Well, if you want something absorbed in the intestines, you may have to put it in like a capsule where the stomach acid doesn't break that substance down until, while it's in the stomach, it passes through, and then that capsule's worn away by the time it gets to the intestines, and that's where it can absorb. So it just depends on what you're looking at. Where do you want something to absorb? What state do you want it to be in? Can it go through the plasma membrane? Can it use an ion channel? So it just depends on a lot of things. But so a lot of times when people look at some of these examples, they're like, why do we care? And it's because, for medication or just certain substances like fatty acids, steroids, fat soluble vitamins, they are going to be able to go through plasma membranes. Carbon dioxide and oxygen can go through those membranes. So having that membrane permeability is really important. So, when we look at these, it's having the ability for these to pass, like oxygen and carbon dioxide to pass through the plasma membrane. That's really important. And we're going to really see how that ties into, you know, surface area in just a second. Because think about like, if you think about like your cell, you know, and you kind of think about that picture where you can see the phospholipid bilayer, you have a lot of the phospholipid bilayer, and then only here and here and here and here in these specific places do you have protein channels. So if you're trying to pass things through relatively quickly in a short amount of time, the preferred route would be to use things that are lipid soluble that can go right through that phospholipid bilayer, because there's a lot more of that area versus individual channel proteins. So when we look at that, the nature of diffusing substance, lipid solubility of that diffusing substance, that hydrophobic or lipophilic. So hydrophobic means water-fearing, lipophilic means lipid loving. So if you're hydrophobic, you're also lipophilic, because if you fear water, you're going to love lipids. So if you have something that's hydrophobic or lipophilic, whichever term you use, those can pass right through that hydrophilic or through the phospholipid bilayer. Hydrophilic molecules, so if you have anything that's charged, so if you have like an ion of sodium or potassium or some other charged molecule, it is going to have to pass through some sort of pores or channels. It cannot go through that phospholipid bilayer. So that's kind of important to know what things are you know, charged or polar versus what's uncharged and nonpolar, because your uncharged nonpolar substances, the hydrophobic kind of substances, can go right through that plasma membrane, whereas if you are charged or polar, so hydrophilic, those molecules have to go through pores or channels. They can't go through that membrane. So here, this is showing you concentration gradient. Here's low permeability and high permeability. So, you know, if you had, you know, when you're looking at those, if you had two different kinds of medications, which one, you know, so you're a drug company, you've made two different kinds of medication. Which one would more likely be the better medication for your patients? Well, the one that has high permeability, the one that can actually get into those cells. The low permeability, you know, you would have to maybe change where it's getting absorbed or something else, like if you have a low permeability, but the high permeability would be the one that would allow easily for it to get into and out of cells. So the last thing that goes into factors that affect diffusion is going to be surface area. So the greater the surface area between two regions, the greater the space is for that diffusion to happen. So the greater the flux that can happen. So if you increase the surface area, it's going to increase the rate of diffusion. So imagine the situation where I had you guys all, like 20 of you guys in the room, walking, bouncing into each other, and until you hit the door. Well, you know, the door is an actual pretty small surface area. Imagine that if I took like the whole wall here on the side where the door was, and I made that a whole, just a big open space there. Now we have a really big surface area, so it's much more likely that you guys are going to move from inside the classroom to outside the classroom without changing the concentration, because the surface area where you can cross has increased. So kind of think of it like that. Surface area is really important when we start looking at how many pores you have available for like certain ions to cross, or if you think about carbon dioxide and oxygen, having a big surface area is going to allow for really the best exchange of those gases. So in real life situations, having increasing that surface area is going to increase that rate of diffusion. So here we can see, here's membrane A, this is membrane B. So here, this would be like, you know, if I opened up the wall or I put a whole bunch of doors that you could get out of there, versus, you know, like what it is now, there's only one door, you know, on the side that faces like the hall where you guys normally come in. So when you increase the surface area, you're going to see that it's easier for these molecules. You see how much, you know, those moved around a whole bunch before one of them made its way out. So having a greater surface area that allows more pores, it's going to allow things to pass into and out of the cell much more easily. All right, so this is from a like really old version of the book, but you can see here they're showing things like sodium channels, potassium channels, chlorine channels, calcium channels. This is showing that the channel can open and close, but a lot of times people think, oh, we have this plasma membrane and there's these proteins that are kind of embedded into it and they let all sorts of things go in and out. In reality, it's not like a one channel lets anything and everything in and out of the cell. They are a lot of times they're specific to specific molecules or specific ions. So you can't just have a channel and it lets sodium in and potassium out and chlorine in and calcium out. They don't function as a one size fits all, everybody can go in and out. They are specific to specific molecules. So when we start looking at these these channels here, they are specific to specific molecules. So here we can kind of see here's a really big molecule. It could not fit through any of those channels, but here that one was able to go through. So if this was a sodium channel and this was sodium, it could pass through. If this is a sodium channel and then we had a green thing here that was potassium, the potassium would just kind of bounce out of there, even though they're similarly sized, would not be able to go through. The potassium can't go through a sodium channel. So we can kind of see here that one wasn't able to go through and that one was. So if this was a chlorine channel and this is a sodium, the sodium couldn't go through the chlorine channel because it doesn't have the right charge or the right size or combination thereof. But when you have chlorine ions, chlorine could go through there. So we can kind of see that you can do things like if you take like Novocaine or lidocaine. So if you've ever had to have like a cavity filled, or maybe you had like a mole removed or something, and they gave you, I understand it's usually lidocaine, and in your mouth it's Novocaine. But both of those use basically a sodium channel blocker. So it's a little substance that covers over those sodium pores. So the sodium pores, even though you have sodium ions, those pores are not available because you kind of like, it's like you lock the door. You're not letting those pass. If you don't have the right amount of sodium that like can rush into cells, when we start talking about depolarization, we're going to see that you won't conduct nerve impulses. So it's not going to allow you to conduct those nerve impulses that are going to send a sense of pain. So, they give you Novocaine and then the dentist can drill on your tooth and you don't feel pain because they've blocked those sodium channels. There's things like cystic fibrosis. So cystic fibrosis is a genetic condition. And what it comes down to is that genetic condition misfolds the proteins that make your chlorine or the chloride ion channels. And so the chloride cannot get in and out of the cell the way that it should, and it causes that really thick, salty kind of mucus that you see in the respiratory and digestive tracts that cause a lot of difficulties in, like I said, we typically think of breathing, but it also affects the gastrointestinal tract as well, because you get this real thick mucus that basically forms with that. And so that whole condition, all of these things that happen that can cause people to have to have lung transplants and different things like that, is caused from basically a misfolded protein that made that chloride ion channel form wrong. So now the chlorine can't enter and exit the cell normally. So just like, once again, you go back to like how proteins are made, what the DNA said, how a protein is made, and a defect there can cause, just one misfolded protein can cause something pretty catastrophic and life affecting. Now they have lung transplants, they have medication that can prolong people's life. So people who have cystic fibrosis can live into, I think, 30s now. But once upon a time, people that had it, if they survived into, to be a teenager, that was a long lifespan for cystic fibrosis. But that's just, just examples of how important these channels are and how specific they are. So like I said, the more channels that you have open, the more of that particular substance can enter or exit the cell, depending on those concentration gradients. So if you close pores or open pores, that's going to affect basically the flux or flow that's happening there. When you look at the surface area, usually when we think about the surface area of like a cell, it's pretty fixed. You can't make a cell really big or small. It's typically the same size. Like I said, you can open and close pores that can affect how things can get in. But you usually, unless you start killing cells, you can't, like, you don't usually alter the size of the cell itself. But if you start looking at things like the lungs, so imagine, oxygen and carbon dioxide, they can go right through that phospholipid bilayer. So they're not going through a protein channel. They can go right through a phospholipid bilayer. So they're going right through your phospholipid bilayer. If you have a condition, you have pneumonia. Pneumonia means that you have water that covers over the surface of, the alveoli, the cells that make up the little air sacs in your lungs. And so when you basically make it thicker, so it makes it harder for that. If you have conditions like emphysema that destroy the lung tissue, instead of having all the alveoli, if you start destroying that, you're going to reduce the surface area of your lungs. So if you imagine like the alveoli like clusters, imagine like a group of grapes, and imagine that's like your lung. If you have emphysema and you start destroying cells and destroying that lung tissue, it'd be like you plucking off a bunch of those grapes on your from your bunch of grapes. So if you look at your full bunch of grapes, your normal healthy lung tissue, there's a lot of surface area there. If you counted all the surface area of all those different grapes, as you start pulling them off and you have fewer and fewer grapes left on your bunch, your surface area is going down because you've destroyed that lung tissue. And so that's going to mean the exchange of gas is not going to be as fast because you have less surface area there with that. So if you, like I said, if you truly destroy the lung tissue, then you take away that surface area. You could have conditions, like I said, that make it hard to breathe, like because of alteration of the lung tissue, even though you didn't fully destroy it. All right, so we will stop here and we will pick up with the next video after this, so that you guys, I'm sure this is longer than I think it is, but I don't want to end up having a like 5-hour long video for you guys to watch. So we'll stop here and we'll pick up with another video in a little bit.